Hello, I am Preeti Rokde and my topic is development of nervous system. Introduction. Nervous system is ectoderm in origin. Nervous system starts developing in the third week of intrauterine life. Central nervous system. CNS starts developing in third week of intrauterine life. CNS appears above the notochord. Formation of the neural tube. Formation of the neural tube begins in the region of fourth and sixth pair of somates. It starts developing during third week. It develops from ectoderm. Formation of neural plate. The development of neural plate begins at the cephalic end and gradually extends towards the primitive streak. Neural plate is located in the mid-dorsal region in the front of primitive pit. The cell of the neural plate make up neural ectoderm. It is also called neuroectoderm. Formation of neural plate. Under the inductive influence of notochord and paraxial mesoderm, the ectoderm overlying the notochord thickens to form the neural plate. Edge of the neural plate becomes raised and more elevated to form the neural fold. The formation of the paraxial mesoderm helps in the elevation of the edge of the neural plate. The central mid region of the neural plate above the notochord remains in situ. The notochord has some blind force with this central mid region. Thus, the neural plate is converted into neural groove. As the paraxial mesoderm thickens, the neural groove depends down between the right and left paraxial mesoderm and converted into neural tract. Gradually, the neural fold approach each and fuse in the midline. This fusion begins in the future cervical region at the level of fourth pair of somates, part of neural tube. At this stage, the cranial two-thirds of the developing neural tube as far as fourth of the somate represent the future brain and the caudal one third distal to the fourth pair of somate represent the future spinal cord. Formation of neural tube. Dorsal view of human embryo at different stage describing the development of neural tube. The amnion has been cut and removed. Formation of neural tube. The fusion proceeds in cephalocaudal direction. At cephalic and caudal end, neural tube temporarily remains open and the lumen of the neural tube communicates with amniotic cavity by way of anterior and posterior neurones. Anterior neuropores closes at day 25. Posterior neuropores closes at day 27. Closure of the neuropores coincides with the establishment of a blood vascular system circulation for the neural tube. Formation of neural tube. Wall of the neural tube becomes thickened from the brain and spinal cord. The lemon of the neural tube covered into the ventricular system of the brain and central canal of the spinal cord. The process of formation of the neural tube is known as neuralization, and the embryo at the stage may be called neurula. Development of neural crest cells. Gradually, the neural fold approach and fuse each other in the midline. As neural folds elevate the fuse, neural crest cell at the lateral border of the crest of the neuroectoderm disassociate from the neural tube. Neural crest are cells are neuroectodermal in origin. They start differentiating during the formation of neural tube in the third week intrauterine life. There appears and elevated edge of neural fold in the region of mesocephalon. Lateral on their extent cranial and caudal. Development of neural crest cell. Initially, they form an intermediate zone in the mesoderm between the surface ectoderm and developing neurotube. Lateral, they divide the midline and are present in the mesoderm dorsal lateral to the neural tube. This cell undergo an epithelial to mesenchymal transition as they leave the neuroectoderm by active migration and displacement to enter the surrounding mesoderm. The migration occurs in a craniocaudal sequence, beginning at the start of fourth week. The neural crest gives rise to cell that forms the PNS and ANS consist of cranial, spinal and autonomic ganglia. Neural crest cell originating in the neuroectoderm of forebrain, midbrain and hindbrain regions. Migrate into the pharyngeal arches and facial region. Here, they form midfacial and pharyngeal arch skeletal structure and other tissues, including cartilage, bone, dentin, tendon, dermis, sensory neuron, and glandular stroma. The lateral of head and neck region of a developing embryo showing migration pathways of cranial neural crest cells into facial region from different part of the developing brain. Formation of brain. The neural tube cranial, the fourth pair of somate develop in brain. The cephalic end of neural tube shows three dilatation. They are called primary brain vesicle, prosencephalous or forebrain vesicle, midbrain vesicle, and hindbrain vesicle. During fourth week, folding of the embryo is also taking place. So, by the end of fourth week, two flexures obvious in neural tube. Cephalic flexure, it located in mesencephalo region. Cervical flexure, it is at the junction of spinal cord. Secondary brain vesicles. During fifth week, 5 secondary brain vesicles develop. Prosencephalon divided in 2 secondary vesicles. Telencephalon or end brain vesicle. Diencephalon. Mesencephalon or midbrain vesicle does not divide. Fourth, mesencephalon, and fifth is myelencephalon. Ventricles of brain. Telencephalon consists of a middle narrow portion and two lateral dilated portions called primitive cerebral hemispheres. The cavities of cerebral hemisphere are called lateral ventricle. Diencephalon is characterized by outgrowth of optic vesicle. The cavity of diencephalon is third ventricle. The cavity of rhombencephalon is fourth ventricle. Thank you for watching my seminar. देख